Hello, hello. Welcome to another edition of the ROM's Google Hangout. And we've got probably the best hangout ever happening today. And we are in the basement of the Royal Ontario Museum. I'm surrounded by ichthyologists who we're going to introduce in a second. And we've got a mystery, like a brand new mystery the world doesn't know about it, that we're going to unveil during this 30-minute talk with our team. And we're not going to unveil it for a little while, so you got to watch. What we are here at the ROM, I'm Dave Ireland. I'm the Managing Director of Biodiversity here at the ROM. And my goal, my job really is to help bridge the back of house, our scientists, our curators, our research and our collections with the public. And at the same time, connect our public and their queries and questions and concerns back to our expertise here at the ROM. Great job, I love it, and I get to work with these people all the time. And we actually have a live studio audience in here today that you can't quite see. Quick yell from the EDC team. Awesome, awesome. EDC is a program here at the ROM. It's called Environmental Visual Communication. These is a brand new cohort of this program, and these students are going to become the next generation of good science storytellers. So they're here learning about fish and ichthyology, and also learning about technology and our amazing tripods and everything that we do here at the ROM. So if I could give a real quick rundown on how this is going to go. Again, the focus of our Google Hangout today is on a mystery. We're going to unveil something, but we want to get to know the team a little bit, so we're going to do some introductions. Then we're going to talk about this mystery a little bit. Then we're going to unveil something here and let you see that. And then we're going to wind up about what we're doing in the future. What does the next steps look like? If you want to participate, the handle really is @ROM Biodiversity. It's our main Twitter feed for anything life science here at the ROM. You can ask, use the hashtag AskROM, ASKROM. This is streaming live on YouTube, on the ROM's website, which is likely the easiest for folks. But if you're a Google Plus aficionado, you can get on there as well and actually type in your questions, and we'll get those. So any questions you want to deliver to the team through Twitter is probably one of the best ways. But if you want to send it to the main handle @ROM Toronto, we'll get that as well. Big shout out to Emmy down at the Field Museum. Emmy, we love you. We want to emanate you. And if you would do us the honor and share this amazing hangout with your colleagues down in the US, that would be fantastic. So, everybody clear? Let's meet our special guest. What a great looking fish team. We have the entire fish team here, and I'm just going to quickly say their names and then give them maybe a minute just to say who you are and what you do here at the ROM. Mary.
I'm Mary Burridge. I'm an assistant curator in Ichthyology. I've been here since 1981. I do a whole lot of things. Do some research on fishes in the Indo-Pacific. I do a lot of gallery work. We teach some Ontario fish identification workshops, gallery work. We wrote the, Erling and I wrote the Ontario Fishes Guide.
The quintessential book on fishes in Ontario. And maybe Erling, after you introduce yourself, tell them about the fish I caught in Algonquin Park as well.
Sure. My name is Erling Holm. I've been here since 1977, so I guess I'm here to talk about the old guy. And my job is mainly involved with the collection and cataloging. And I'm very excited about our 100,000th lot that we're going to have right now.
And another guide.
Yeah, Fishes of Toronto. I was involved with that committee that produced that book. And
yeah, all fish.
All fish. Dave caught a fish, which he wasn't quite sure what he thought who he knew what it was. He showed it to one of our guards, Chris, who used our guide, and told him it was a fall fish. So fall fish is Ontario's largest minnow. It can get up to, how big can it get?
At 50 centimeters, they said.
All right.
And mine was about 49.
Oh, right.
You're almost there. Almost there. So let's just do the back here quick, Sarah.
Hi, I'm Sarah Steele. I'm a graduate student at the University of Toronto and working with Hernan and Lopez-Fernandez, who we'll meet in a second. I've been a graduate student for about 2.5 years here at the ROM, studying fishes South America.
And quickly, Sarah, this is huge. Sarah's you're a University of Toronto
Yes.
student,
Yes.
but you are at the ROM. How does that work?
Yeah, so officially I'm at University of Toronto, but a lot of the curators here give opportunities for graduate students to actually do the research out of the museum. So studying biodiversity, conservation, I'm studying evolution of these fishes, so it gives us a great opportunity to work with natural history. So
And do you get to go south of the border?
absolutely.
Where were you last?
Last I was in Guyana and Suriname, so that's in northeastern South America, catching fish there.
So, quick message. If you're one of Hernan's students, you get to go to Guyana and Suriname. That's pretty cool.
Maybe.
Maybe. I think it's up to
A lot
him.
of applications.
And at the back here, Mark.
Hi, I'm Margaret Zur, and I'm the technician in the Ichthyology department. I've been here since 1982. And I sort of do a little bit of everything. I kind of support staff for everybody.
And I also see you from time to time, Mark, up in the Shad Gallery of Biodiversity near our amazing aquarium. What do you do up there?
I sort of run up once or twice a week and make sure everything is running well, and I feed the fish, and I talk to visitors about our wonderful aquarium, and just keep an eye on things.
And once a year, Mark sends an email to us saying, we need more budget, we need better fish.
We do.
This
We
is the voice.
We actually do need some hermit crabs. I don't know what's happening.
We'll go offline about that a little bit. And really the lead of this lab, Dr. Hernan Lopez-Fernandez, is our curator here at the ROM, and he leads our ichthyology department. Hernan, tell us about yourself.
Well, hi, Dave. Hi, everybody. So I'm Hernan Lopez-Fernandez. I'm the freshwater fish curator at the museum, and I am also an associate professor at the University of Toronto in the Department of Ecology and Evolutionary Biology. So essentially what we do together here is to use natural history collections, in our case, the fish collection, to address questions that range from taxonomy, as in describing new species, to understanding the evolutionary process that originated what is actually the most extraordinary diversity of vertebrates around the world. Fishes, there's well over 32,000 species of fish described, and we study a modest portion of those, particularly concentrating in South and Central America. Our research also involves a little bit of conservation biology and a lot of field-based exploration and discovery of new biodiversity, because all these things are tied to each other. We cannot conserve things that we don't know exist, and when we start learning that different species occur in different parts of the world, then we can start piecing together the history of how they evolved into such a diverse group. And in the particular case of South and Central America, which we call the neotropical region, we have the largest concentration of freshwater fish diversity. We don't know exactly how many, but we estimate close to 8,000 species, many of which are still unnamed. And part of our work is to try to go to remote places that haven't been explored carefully enough and determine whether there's as yet undetected or unnamed biodiversity. Learn a little bit about the conservation status of both those species and the regions where they live, and then add that information to our growing collection and information from other collections around the world to try to understand how such an incredible diversity of freshwater fish originated in this particular region of the world.
And so, Hernan, you're the current curator of ichthyology. Are you the only ever curator of ichthyology?
I believe I'm the 8th curator of ichthyology
In how long?
in 100 years.
100 years.
So the ROM, as you know, opened its doors in 1914, and since then we've had 7 or 8 different curators. I can't believe I can remember that. So one interesting aspect of that is that one wonders what exactly is a natural history collection, and what is it that we keep. And in many ways, what we have, where we work, and what is the focus of our research is driven actually by the curators. So most recently, I shared the position of curator with now emeritus curator Richard Winterbottom, who spent, 30 years at the ROM studying the origin and diversity of coral reef fishes in the Indo-Pacific Ocean, of which we now, because of Rick's work, have one of the most important collections anywhere. And that's the key about the curatorial work that we do, is that we focus our efforts on a particular region or in a particular set of evolutionary conservation questions. And then we use the information in the collection to piece together the stories about that fauna. And when we don't have enough information, we go out in the field and we try to find it ourselves, which is what keeps the collection growing and relevant over time. Which
and is a great segue, because I think we're going to tease out a little bit of that mystery now. So imagine it, 8 curators, 7 curators, 100 years, each with a focus: Indo-Pacific coral fishes, cichlids from South America, Canadian fishes. Didn't we write the book on Canadian fishes? Dr. Crossman. So all over the world, these different niche pockets. We have amassed. I mean, Brian, show them the jars, and I mean, he's standing behind jars and jars of fishes. We have Erling. How many fish are currently, plus or minus 5,000, whatever? How many fish are in our collection?
Well, we're over 1 million, for sure.
Wow.
In about 100,000 lots.
Okay, so this brings me to a point, and I'm going to throw it out to anyone. What the heck is a lot?
all right. So I can help you with that one.
Okay.
So a lot, for most people, is a jar full of fish, right?
For
So-
most people, or
For
for
most people. For us, there's a little more nuance to that. So a lot, technically speaking, is a container that has all the individuals of one species collected at the same time in the same place anywhere on the planet. So essentially, a lot is sort of the basic currency with which we do research in fish biology and ichthyology. So it may seem like a humble container, but actually, when we do things right, this lot contains an enormous amount of information that we can use in many different ways and then integrate into one coherent story about those fishes and whoever is related to them. So for instance, this lot has a label over here that says that those fishes were collected in Guyana. They belong to a species called Guyanacara dacrya, which incidentally was described by one of my students and myself a few years ago. And inside the lot, there's these specimens that have a wealth of information associated with them. So for each one of them, or for a representative number of the specimens in each jar, we have a collection of tissues, basically a little sample of flesh that's stored in special conditions so that then we can access the DNA and sequence the DNA. We use the DNA to do many different things, but for instance, one of the most important ones that we work on a daily basis here is reconstructing the family tree of these fishes, which sounds kind of like an arcane thing, but when you can reconstruct the family tree of a group of fishes, you learn who is more closely related to whom, and actually you can combine those family trees based on DNA with a fossil record. And if you have ages of fossils that you can place in the family tree, then you can use a bit of mathematical and computational magic to actually calculate the age of every one of the species in the family tree to which that fish belongs. So all of a sudden, from the little bit of tissue that we collect in the field, we learn who's most closely related to whom, how long ago each one of those species originated, and that gives us sort of a roadmap to the evolutionary history of all those fishes. And then we can keep exploiting the information in the jars to look at things like, for instance, we can compare what these fishes eat. We can always look at what they ate by looking at their stomachs or by looking at chemical properties of their tissues. We can learn about their reproductive biology. In the field, when we're collecting them, we gather all this standardized information about their habitats, about the type of water where they lived, whether they came from habitats that were pristine or affected by human activities. We also have detailed geographic information, so we can put all this stuff together to ask questions as, for instance, why is it that this particular group of fishes evolved some million years ago and became a specialist in eating insects from the bottom of rivers, and how did they do that? And we can use the specimens to look at, for instance, what we call the biomechanics of feeding or the functional morphology of swimming. We can compare the evolutionary pathways that led to different ways in which these fishes swam, or why some of them are really big or really small, which is the subject of Sarah's PhD
So
dissertation.
you play with lots of lots?
I do, yes.
So big take-home message here, folks, is this one jar. And Hernan had about 10 different topics that he could draw from one jar. And just to give a bit of a shout out to the team, if you come to the ROM until September or early October, they curated an exhibition. It's a temporary exhibition as part of our centennial, and the stories that Hernan just told are graphically represented on the wall in the Shag Gallery Biodiversity. You can take your time, you can learn all about it, and it's a really nice display. So if we have all of these lots, what are we celebrating today?
Well, today is a special day.
Okay.
Because, as you know, the ROM is 100 years old.
Yes.
And today we're going to mark a landmark, sort of a shout out and happy birthday celebration to the museum by actually cataloging our 100,000 lot.
Cool.
Which means that we've reached an important landmark in the history of the fish collection and in the history of the ROM. And also we're going to celebrate it, because I mean, it's not any number. So we're going to celebrate it by cataloging a fish about which we know nearly nothing.
Woo!
So what
New species.
it's not just a new species, it's a new genus.
Whoa.
It's a fish that doesn't have a name on two accounts. We don't know what genus it belongs to, because as far as we can tell, it's the only member of that genus, and nobody knew about it until 2008. I'll tell you a little more about that in a minute. And of course, because it's a new genus and nobody knew about it until so recently, it also doesn't have a species name. So we've been working on trying to describe this new species, which has been kind of difficult because, like I said, we know very little.
Quick tangent. Can we name it after one of us? How does that work? Who is it going to be Hernan Lopez-Fernandez Ichthyology, or?
No, that's not the way it works.
Okay.
So first of all, I can't name a fish after myself.
Oh, okay.
I don't think that's allowed, and even if it were, it's not something that people do.
Okay.
You can name species after people. Usually that is done after people that have contributed significantly to either the discovery of that particular species, because say they were the ones that collected it in the field, or people who have a history of contributing to the field and want to be recognized, which is not to say that people recognize other people by naming species after them for all sorts of reasons. But in general, my particular preference is to try to name the fishes after attributes that describe them.
Fair enough.
So
had to ask.
Absolutely, it's actually a frequent and important question.
Okay.
So anyway, the story of this 100,000 lot is actually really cool, and it's a bit of an unexpected story here at the museum. In 2008, we had this totally serendipitous invitation to go to a very remote part of Guyana that was very poorly known. We knew that it had some strange fishes because long before a biologist had been there and had collected a few specimens, and those few specimens turned out to be 3 different genera, all belonging to that strange river. But the place is so remote that nobody went back again. I'm talking about, I think, the 70s or early 80s. And then for the first time in 2008, we had this opportunity to organize a formal expedition to explore what the fishes of this upper Mazaruni river basin in Guyana were. And this place is really strange because it's perched atop the Guyana Shield in northern South America. It's one of the oldest geological formations on the planet. It's very isolated from everything else. So we were there a couple of times since then, and we've found a lot of new things. And one of them is this fish that we actually nicknamed originally Zigzag because of the color pattern that it has on its sides. And we know very little more than that. So now we've been working on it for a few years. And because we're in the process of describing, the first thing that we need to do is actually identify a number of individuals of that species that are representative of what defines the species, what makes it different from everything else, so that from the moment that we name the species on, every biologist who wants to identify their fish can come and compare them to this particular type series, as we call it. So what we're going to do today is essentially 2 things. This little fish that's in the little aquarium, that's a very special individual. That's
So just quickly, folks, what we're seeing, and you can't see.
I don't know if you can see it,
but what we're seeing is the holotype.
the holotype.
So the original, like the one. And so what we can do, Hernan, because you're going to take some pictures of this, right?
Yeah.
If our viewers out there, and I'm going to stop for a second and ask if there's any questions, but if our viewers out there want an image, want to see this, we can send it to them, right?
Absolutely. Yeah.
So if you want to see this image up close, see this brand new unnamed species, genus of fish, give us a call. Real quick from our studio audience or anything on Twitter, Ryan, is there any questions quickly at this point? Round of applause. All right. Okay,
So what we're gonna do now is photograph the holotype, and this fish is special because it will, from the moment that the species description is published on, be the key reference for anybody to come and see, okay, so is the fish that I have this one that is already described, or is it a different thing? And the other thing that we're going to do is designate a lot of paratypes, which is going to be our 100,000 lot. And paratypes serve the purpose of spreading this exemplars in various collections, so that we ensure that, on the one hand, they are accessible to ichthyologists everywhere for comparison, but also that if anything were to happen to the holotype, there's still specimens somewhere else that can be used to go back to the original description, so that people can still have a good idea of what that species
And
was
didn't you
supposed
tell
to
me
look
just
like.
before we went on that this actual individual has to go back to
To
Guyana?
Guyana, exactly. So legally, the countries where species originate
Yep.
should have their fishes back. So when we describe
That's a
a
very important point.
new species from Guyana, we are actually obliged to send the holotype back to the museum at the Center for the Study of Biodiversity at the University of Guyana, which right now holds the fish collection for the country.
Okay.
So what we do is create a series of paratypes, lots with other specimens that are also representative of the species. We keep some of the ROM, we send some to other museums with which we collaborate,
Yeah.
so that we ensure that those specimens are available for the scientific community to make their comparisons, and also to make sure that they don't get damaged or disappeared. For instance, during the World Wars, many museums suffered losses of their type materials
Right.
because of bombings and things like that, and many types that were described before that time have been lost. So now we're trying to ensure that.
I'm just going to ask a quick question over here of Erling. So this fish came from Guyana. It's brand new. It's 2015. It's an unknown genus. Could I go to Algonquin Park and find a new species?
Unlikely.
Unlikely.
Yeah.
The Arctic?
Well, actually, with new DNA work, it is possible that you can find new species that just look the same, and we haven't been able to differentiate. So there are, say, of the sticklebacks,
Yeah.
there are two genetic forms that look almost identical. They're very hard to separate. So it is possible, if you can find between those two species some difference that you can see, then you can start describing a new species from Ontario.
And so the point I'm trying to make is that generally we know the Canadian
Generally,
fauna.
we know it. Yeah.
And so just to emphasize this point, where Hernan and his lab is working is kind of the epicenter
Right.
of cichlid fish diversity in South America.
Well, not just cichlids. Cichlids is a family that we do
Yeah.
a lot of research with. But no, it's actually true for every family of fishes in South and Central America. So it's one of the regions of the world that still needs a lot of work in discovering.
And where would two other regions be?
Southeast Asia,
Southeast Asia.
particularly the Sunda Shelf and New Guinea and places like that. A lot of Africa remains full of undescribed species, and there are people actively working in both regions.
So we're a regional museum. We're called the Royal Ontario Museum, but we work everywhere.
everywhere, literally.
Everywhere. And 100 years ago, we started in the Humber and the Don, and where we're bioblitzing nowadays. We started here in our local watersheds, built out, and now we're in these hot spots because of these reasons.
Right. Absolutely.
Cool. So are we
So
photographing
yeah,
it?
we're photographing the holotype, which shouldn't take too long.
And dun, da, da, da. So everybody out there in internet land, do we have any idea what we're naming it at this point? Is
No,
it
and even if we did, I couldn't divulge it.
secrets?
Yep, it's part of the rules of naming. these
So
things.
Sarah, as a graduate student, what is the penultimate experience? Would the penultimate experience for a young fish graduate student to be to find and identify a new species?
It could be, and I have done it before, and it was one of the most exciting things I've ever been involved with. So, yeah. A lot of people are very interested when we talk to them.
And was this under Hernan's tutelage?
Yes, absolutely.
Awesome. And how many, just maybe you don't know, I don't know, how many fish has this lab over 8 curators described?
We're about 126. We've got 126 photographs.
Kill me into the camera here. How many species of fish?
126. Started, as Dave said, started very locally. We've got a few from Canada, subspecies and species. And then as the years progressed from 1914, we spread out. We did the Arctic, we did some South American, and then we did a fair number from Southeast Asia, and we have a lot from the Indo-Pacific area from Wonderbottom. So 126 or even more, because every year we're doing half a dozen or so more. So.
And is there kind of subtle competition amongst the different labs at the ROM on this level? Like,
Not really, they can't compete with us. Maybe entomology could.
I just heard a stat the other day, and maybe Emmy and others know this, but there's a group of animals called the parasitoid wasps,
Yeah.
and they think there's about 60,000 of them described, but there could be 2 million out there.
Yeah.
So entomologists kind of have one up on our
Them-
vertebrate
Yeah,
friends.
they might, but I mean, insects. They're not.
So can you tell there is a bit of competition? Yeah.
Fish raw.
Okay, guys, so unfortunately, time flows. We only have about 3 minutes left, and I'd like to just go through all of you. Oh, is there one more thing to?
Well, let's do it.
Oh, let's do it, yeah. So this is how a lot is made.
What we've done here is we've chosen 3 specimens that are particularly well preserved and informative as to what this species looks like. And we're going to take them into becoming a new lot. We've already created the label for that. And this is going to be ROM 100,000 for the
There it is.
There you
Is
go.
the name on it? No,
Nope.
there's no name.
It's a nickname.
It's a nickname. Can we, I know sometimes scientists are back and forth on this, but kids, families love it. Are we going to nickname this species like we did the dinosaur, or do we even want to go there, Hernan?
Nope.
Okay, we're not going to go there. But if you want to nickname the fish, you could tweet us. So we only have a couple minutes left. This was remarkable. We've got a
There you
new
go.
100,000th lot of fish. Amazing. So it's a perfect segue, and I'd like to just whip through the 5 of you. It kind of feels monumental, maybe climactic. What's next? Like, we'll start over here with Mark. If we could take, really quickly, Mark, if we could take something into the future, Fish Lab, what would you like to see happen over the next 20 years?
Well, I'd like to see us continue on this path. I think we're doing really great work. Hernan is bringing in lots of fish from South America. The kids are learning a lot. The students are really engaged. And everything is, I think, working well.
Awesome. Sarah, you want to
Yeah,
graduate?
yes, yeah, finish the thesis. But I think one of the most important things being involved as a student here at the ROM is connecting with the public and just teaching them about specifically what I do as well as what we all do here at the ROM.
Awesome, awesome. Erling? Rewrite Book of Canada?
Yeah, we're certainly going to continue revising our book.
Yeah.
And Erling is working on keys for the freshwater fishes of Canada that is going to come out, right? The big Bible of Scott and Crossman that was put out in 1976 here at the ROM, and Erling is now making new keys because there are so many new species that have arrived in Canada, invasive species or species that are disappearing. So Erling is rewriting keys for that, which could come out in the next couple of years, hopefully.
And in 30 seconds, Erling, 30 seconds, what do you and Mary do every spring in April in Guelph?
Oh, we put on some fish identification workshops. This year we had about 130 or 40
At least.
people.
Did everybody
Mostly.
pass?
Yeah,
We're serious here.
It's a pretty intensive, tough course.
And they're
So,
Ontario fish, workshops for Ontario.
Ontario fish.
Yeah.
It's a really great
Even
course.
though they have to know only about 150 species.
Only. Yeah.
And if you're interested, yeah, you can contact the Department of Natural History at the ROM and you can sign up for next year. We do them every spring.
Awesome. And Hernan, last word, 30 seconds. Where are we going?
I hope that we're going towards understanding the diversity of fish is much better than we do right now. And thanks to our work down the road, when I get to retire, those fishes are still there. And our work helped keeping them in good shape.
Awesome. He's already talking about retirement. He's got like 30 years left. So folks, again, the ichthyology team here at the ROM. Wonderful job. And wherever it is, our 100,000th lot of fish. Oh my God, this is a bit scary, but there it is. And a quick shout out, everybody who's listening. A part of this team, plus about 40 other members of the ROM community, will be in the Dawn watershed at base camp at the Ontario Science Centre on June 13th and 14th. And the goal of this 24-hour BioBlitz, which is arguably the biggest one in Canada, is going to be to help our scientists connect with the public in a real physical way, a hands-on way out in the field. We still have space in some of the guided blitz programs. If you Google BioBlitz, we come up right under Wikipedia before National Geographic. So you can find us there. Hopefully sign up again. That's June 13th and 14th at the Ontario Science Centre. On behalf of the EVC program, the ichthyology team, signing off here from ROM Biodiversity. Thanks, everybody.